we'll be starting with today's class, that is transcription, the next part of central dogma, as was proposed by Crick in 1955. And we are moving on from replication to transcription, and the books are more or less the same, which I referred for replication. And now we've come to transcription. Now, when we talk about transcription, first let us understand the basic difference between a DNA polynucleotide and an RNA polynucleotide. Both DNA and RNA have the basic units of a nucleotide, okay? Now, the sugar in case of DNA is deoxyribose, but in case of RNA, as you can see, where did my cursor go? The 2' end, so it's 2' deoxyribose for DNA, but for RNA, as you can see at the 2' end, there is an oxygen. So it's basically a ribose sugar. So it's a ribose sugar. Bases, in case of DNA, we have A, G, C, T. In cases of RNA, the T is replaced by U, uracil. So we have thymine, we have uracil in place of thymine. Now, in case of RNA also, a single unit of nucleotide is joined by another unit of nucleotide by a phosphodiester bond. So the nucleotides in case of RNA, in a very similar manner as to what we saw in DNA, are joined to form a long polymer. Now comes a very big difference between DNA and RNA is that DNA in nature usually is double-stranded, but RNA in nature is usually single-stranded. So RNA is basically single-stranded as compared to DNA, which is usually found to be double-stranded. So the main structure of DNA is the DNA double helix, or the Watson-Crick and Wilkins model of DNA double helix. But in case of RNA, since RNA is single-stranded, it is observed that they exist in a lot of secondary structures. I'll show you one figure later on. And stability, DNA being a double-stranded molecule, as you can understand, its stability is greater than that of RNA, which is a single-stranded molecule and is prone to digestion by endonucleases present in the cytoplasm. So RNA is prone to digestion by endo and exonucleases present in the cytoplasm. So it's easily degraded, while DNA is much more stable because it's a double helical structure. So these are the basic differences between the structure of a DNA and a RNA. Now, so just this is again a picture of, as you can see, what I've showed you is a picture of the central dogma. So what we have done, what we have completed is, we have completed a DNA opening up and a new DNA being synthesized on the older DNA as templates. And we are getting 2 copies of DNA, and the process is replication, and the enzyme is DNA-dependent polymerase. The process is an autocatalytic one because on deoxyribonucleotides are being made. So it's an autocatalytic one, and the enzyme is, the DNA itself can replicate its own daughter strand, and we saw the machinery. It needs a host of partner enzymes to coordinate the process. Now, when we think of transcription, we have to assume here also that the DNA opens up, because unless and until the DNA opens up, how will the enzyme get access to the single-stranded DNA? So the concept in case of replication, we saw that DNA forms a replication bubble and replication initiates. In case of transcription also, the DNA opens up for some around 17 base pairs. And here, DNA-dependent RNA polymerase, look at the enzyme. The name has now changed. It's a DNA, so it can recognize a DNA and can accordingly join the complementary bases of a ribonucleotide. So it's a RNA polymerase. So it's a DNA-dependent RNA polymerase that recognizes one of the DNA as its template and then starts copying an RNA from that template. So it's a heterocatalytic. As you can see, the mother substrate is deoxyribonucleates, and the daughter that is produced from the substrate as a product is a ribonucleotide. So the two molecules are chemically different. One is a deoxyribonucleotide on which a ribonucleotide is being produced, is being made, constructed. So this is a heterocatalytic process. And so we have this process known as transcription. So the DNA is transcribed. That's how we say it in molecular biology. And what we get is our RNA. Here the picture of mRNA has been shown, but we get an RNA molecule. And if you remember, in my initial classes, I had said, those segments of DNA capable of making only an RNA molecule are known as functional genes. But if you look into this picture, some RNA molecules are again decoded within the ribosome to be translated into a polypeptide. So some RNA molecules, which are known as messenger RNA because they carry the message of the DNA within them, are then decoded in the decoding center of the ribosome to give us a polypeptide. So these RNA are actually the intermediate product. The final product is the polypeptide. So segments of DNA where we get polypeptide as the final product are known as structural genes. So I just recapitulate with what I started in my first class. So this is the basic process of central dogma. Later on in 1980s and 90s, when this enzyme reverse transcriptase was discovered, and the one we studied when we did telomerase, I said that enzyme is a reverse transcriptase. So when this enzyme was discovered, it was observed that this is an RNA dependent DNA polymerase. If you remember the telomerase, it uses 3' DNA as a template, and it synthesizes an RNA molecule on the information based on the information provided by that template. So this is an RNA dependent DNA polymerase, and this can synthesize an DNA from a RNA. So we are not getting into this. We will now restrict ourselves to transcription, where the enzyme used is DNA dependent polymerase. So now let's look into the various categories of RNA that can be produced from a segment of DNA. The traditional standard categories that you are all aware of, you have heard of them, are the messenger RNA, the ribosomal RNA, and the tRNA. The messenger RNA, the tRNA, and the rRNA form a very integral part of the translation machinery. So we'll be doing the translation machinery. All the three RNAs are needed so that the message in the mRNA can be decoded by the ribosome to produce a polypeptide. These three classical RNA molecules are found in both prokaryotic and eukaryotic cell. But there are some RNA molecules which are speciality of the eukaryotic cells. And as you can see, it includes the pre-messenger RNA, the small nuclear RNA, the small nucleolar RNA, the small cytoplasmic RNA, microRNA, small interfering RNA, and PV interacting RNA. A lot of these RNAs are derived from mother RNA molecules, and most of them play a big role in gene regulation, in gene silencing. So we'll not be getting into the mechanism of action of these molecules. For this particular topic, we are now concentrating on that area as to how on a DNA template, a messenger RNA is synthesized by a DNA dependent RNA polymerase. As I said, I talked about the various categories of RNA found in the prokaryote and the eukaryotic system. These are the traditional and the classical RNAs observed in the pro and the eukaryotic system, and look at the location as to where they are found. Ribosomal RNA, cytoplasm. It forms an integral part of the ribosome itself. Messenger RNA, produced in the nucleolus, but it is transferred from the nucleolus to the cytoplasm, and it carries the genetic code for proteins. Transferred RNA, they are present in the cytoplasm. As I said, they form an important component of the translation machinery. They help to incorporate amino acids into polypeptide chain. Now we see that these are some speciality of the eukaryotic RNA molecule, eukaryotic cells. So these RNAs are found on the eukaryotic cells, and most of them, I'm not getting into details of this, most of them are associated in some sort of gene regulation or gene silencing. So this is a more detailed function of the different RNAs. Let us restrict ourselves to the mRNA, because we'll be studying how an mRNA is synthesized on a template DNA. tRNA will be coming, we'll be doing a detail of tRNA in translation, so I'm not taking up tRNA and rRNA right now. And another RNA that you have already studied is the telomerase, enzyme telomerase, which is involved in the replication of the ends of chromosomes or telomeres. Okay. And now coming to this mechanism. So if we look into this mechanism, so now before getting into the mechanism of transcription, the question arises is how transcription, replication and transcription differs. So I'll first ask you people. So as you people have already and beautifully stated the differences, I'm just summarizing the differences between replication and transcription. Transcription is chemically and enzymatically very similar to DNA replication, but involves enzymes that synthesize a new strand of nucleic acid, which is basically a ribonucleotide strand, to a DNA template strand. Now, in case of transcription, the enzyme that catalyzes the synthesis is RNA polymerase. And the RNA polymerase does not need a primer to initiate RNA synthesis. So this is one advantage the polymerase enzyme has over DNA polymerase. In case of transcription, we do not need a 3' end or a primer to give us a 3' end, which was an essential feature required for DNA polymerase. Third, this has been said by you that RNA product that is produced does not remain base paired to the template strand. Rather, as you can see in the picture, the RNA product is remaining hanging. You can see it here. So the RNA product almost hangs, and only a few nucleotides remain attached to the template DNA. Then transcription, even though accurate, is less accurate than replication. Replication is much more stringent, and in replication, one mistake occurs in 10 lakh nucleotides. So the rate of mistake in case of replication is 1 mistake per 10 lakh nucleotides, while in case of transcription, it is almost 1 mistake in 10,000 nucleotides. So the transcription, in case of transcription, the error is more. And finally, in case of transcription, the machinery copies only certain parts of the genome and can copy that part of the genome multiple times, 1 to several thousand copies of that part of the genome. But in replication, the entire genome is to be copied once, only once, during one cycle of cell division. So these are the standard differences between replication and transcription. And if we look into this particular basic concept of how transcription starts, you can see there is a replication bubble form, And as I said, there is no need for RNA primer. The polymerase can recognize the DNA nucleotide and RNA polymerase can start synthesizing the RNA, daughter RNA immediately, and the nucleotides are added in the 3' end, because as I said, the chemical mechanism of addition of ribonucleotides is very similar to one that we saw in the polymerization of DNTPs. So the 3' end is the growing end, because the 3' end contains the OH, and that oxygen actually produces a nucleophilic attack for an incoming RNTP, ribonucleotide triphosphate. And the 5' so it's only around 17 base pairs, 10 to 17 base pairs that remain attached with the DNA, mother DNA template, and the remaining of the strand, as you can see, hangs. And one more feature, as you see, the machinery proceeds. So initially we have a replication bubble, around 17 base pairs of DNA opens up here. And as the bubble proceeds, you can see the DNA behind reanneals and the DNA in front unwinds, and this machinery as a whole is proceeding. So this is basically a concept of how transcription occurs. And one more thing, the important thing is that since only one strand is being copied, so it's not much of a difficulty in here as compared to the replication machinery, where we had two strands being simultaneously copied and the two strands were in two opposite directions. So that was a difficulty in case of replication, which the transcription machinery does not face. And there are 2 to 3 more things I want to show you from this picture. So when the DNA opens up, look at the picture. Let us concentrate the DNA. So we have a mother DNA, which is pink in color. And we have a small RNA, you can see here, which is green in color. So when the DNA opens up, the polymerase recognizes any one of the strand as the template. So how does a polymerase recognizes? There are certain sequences upstream of that strand, which the polymerase recognizes, sits on those sequences and recognizes any one of the strand to be a template. For this particular figure, you see it has below strand. We have two strands here, one in the upside and one in the downside. The downside pink strand has been recognized by the polymerase as the template strand. And it is on this template strand that another thing is that the recognition, obviously, since this is a polar synthesis, only the 3' ends grow. So the DNA template which will be serving as a mother will be the 3', 5' DNA. Mind it, the polymerase will recognize the 3', 5' DNA as a template and will now start synthesizing RNA in the 5', 3' direction. However, there are other sequences also present which helps the polymerase to identify the template strand. So once the template strand has been identified and the polymerase starts synthesizing this small RNA molecule, as you can see here. Now, this RNA molecule is contains the complementary basis of this mother DNA strand. So if this pink one is the mother, the green one is actually complementary to the pink one. Now, if you look here, the non-template strand was initially complementary to the down strand. So, and the green one is also complementary to the down strand. So if the pink one is complementary to the down strand and this green RNA is complementary to the down strand, so actually the RNA is copying the basis of this strand, which is the non-template strand or the coding strand. For example, let me give you an example and explain it here. pen. Suppose here we have these bases. Sorry, just a second. We have these bases here. A, this is DNA. So we have A, T, G, C. Suppose there are 4 bases here. So the complementary strand will bear T, A, C, and G. So this is the sequence of the complementary strand. Now when the RNA is base pairing with this strand as the template DNA, so what will be the sequences in the RNA? So it will be A, in the RNA we are having this sequence. Oh, A, let me write it again. A, T, G, C. We have here T, A, C, G. And now let's look into the RNA molecule. T, so it's alternate. T, A. For A, it will be T, but RNA has uracil instead of T, so it's U. For C, it will be G for the RNA. And for G, it will be C. So you can see the sequence of the base is now similar to the sequence of the bases present in the non-template DNA. So in the non-template DNA, the sequence was ATGC and we have AUGC. So keep in mind that in this transcription bubbles we have, when there is a template strand to which the RNA has, primase has bonded and is synthesizing the RNA based on the template. So actually the RNA is copying the non-template strand. Hence, the non-template strand is often referred to as the coding strand or the sense strand, because the bases of that strand are being actually coded into the RNA. So it is the coding strand or the sense strand, and the template strand is often referred to as the non-coding strand or the anti-sense strand. So after having this basic concept that we have a DNA template strand on which the RNA is synthesized, and the opposite strand is the non-template strand and is actually the coding strand and the sense strand. Now we move on to the basic concept of prokaryotic gene expression. To start with prokaryotes, you should be thorough with how a prokaryotic cell looks. If you imagine a prokaryotic cell, it's a cell with a central genome present in it. It does not have a nuclear membrane. You people have studied that prokaryotic cells lack nucleus and a nuclear membrane. So they have a sort of a genome that may be a DNA, that may be an RNA, and surrounding, and that structure is present in one part of the cell and is roughly described as the nucleoid. So they do not have any cell membrane. Sorry, they do not have any nuclear membrane. So the DNA is present in the cytoplasm, and in the cytoplasm, the RNA is synthesized. And in the cytoplasm, the RNA is then decoded to produce the polypeptide. So this whole machinery of central dogma is operative within the cytoplasm itself. But just to have a look at the eukaryotic system, the eukaryotic system has a defined nucleus, and thus there is a nuclear membrane, and there are pores on the nuclear membrane known as nuclear pores, which help in transport of molecules from the nuclear plasm to cytoplasm or vice versa. So this is the first difference between a prokaryotic cell and a eukaryotic cell. With respect to the machinery of central dogma. That is, everything in a prokaryotic cell, the whole process of central dogma is occurring in the cytoplasm itself. But in case of eukaryotes, replication and transcription is occurring within the nucleus. The translation is occurring in the cytoplasm. So there is compartmentalization. Now again, moving on to this particular slide. So we have a DNA, as you can see in this picture. Just let me see where my cursor is. So we have a DNA, and you can see there is a unit, something written as regulation of transcription. So there is a segment of DNA which will actually tell the enzyme which is the coding, which strand is to be, which strand is to provide the template strand, and which strand will provide itself as the coding strand. It is at this level that the polymerase sits. So it's at this level that the polymerase sits, and then the polymerase starts synthesizing the DNA. So as you can see, this segment of the DNA is known as the coding region, because the information in the non-template strand or the coding strand is copied in the mRNA. Mind it, just one difference that occurs in the DNA, all the T's are coded as U in mRNA. So we have a segment of DNA which will provide the platform where the enzyme polymerase sits, identifies the template strand, and accordingly codes the non-template strand. And the polymerase moves, and finally, again, there comes a point where there are signals, and in these signals, these signals give polymerase the message that the coding region of DNA is over. It is now time for you to leave the DNA, and so the polymerase opens up. This particular structure, this is a transcription unit of DNA. Note that the region which is known as the regulation of transcription area, the region which is known here as the regulation of transcription area, we do not get any RNA from it. So the RNA is coded from the region which is known as the coding region. And as you can see, we are getting an RNA copy, which I have drawn with a green one. And you see, so they have shown the RNA copy here. So the RNA starts from here, as you can see. And see, the coding region is over by here. But it has been seen that the terminator region of the DNA, from where the polymerase is given the message that you leave, polymerase is given the message that you leave the DNA, that portion is also into the RNA. So this portion where the polymerase comes and sits and the regulation of transcription occurs is written here. This is known as the promoter elements in case of DNA. And this portion where there are signals to the polymerase to leave the DNA is known as the terminator element of the DNA. So when the DNA is coded into an mRNA, we have a nascent mRNA, which is known as the primary transcript. And it is this primary transcript which is then decoded in the ribosome decoding center to give us a polypeptide. Note, one thing to be noted, that the 5' end of the mRNA signifies the N-terminal end of the polypeptide. The 3' end of the mRNA signifies the C-terminal end of the polypeptide. So in a prokaryotic system, what you saw is that the polymerase comes and sits in the promoter region. copies one strand of the DNA, which is a coding strand, and then leaves from the terminator region. Point to be noted is that the promoter is never copied in the primary transcript, but the information of the terminator is copied in the primary transcript. And from the primary transcript, we get a polypeptide. This whole machinery of central dogma, that is a flow of information, is taking place within the cytoplasm itself. So this is a nutshell of how prokaryotic gene expression takes place. When we compare this scenario with that of a eukaryotic scenario, we have a similar structure here. So we have promoter elements here also in case of eukaryotic system. So the polymerase comes, sits on the promoter. In fact, the promoter sequence tells the polymerase which strand to be copied and which strand is act as the template. Then we have the coding region. So the mRNA is made from the coding region. And as you can see, the mRNA is copied, and we have a terminator region. So the termination information is produced in the mRNA, and the terminator region is also copied, just like in the prokaryotic system, in the eukaryotic system also. The information present in the terminator region is also copied into the mRNA, but the coding region, we started from here, the two arrows. Sorry. Erase. So we have this coding region. So now when we see the mRNA and the protein, you see the protein is made. So this is the primary transcript. Now in case of prokaryotes, what did you see? The primary transcript was copied into the polypeptide. As I said, there is no nucleus in the prokaryotes, and everything is occurring in the cytoplasm itself. But in case of eukaryotes, we are now having an issue because we have a nucleus. Now, if the primary transcript is allowed to enter into the cytoplasm in this manner, there are exonucleases present in the cytoplasm that will chew up my mRNA. I need to protect the mRNA. So I am sending it from the nucleoplasm, sending it from nucleoplast, from an enclosed structure to the cytoplasm, where there are so many enemies of the mRNA. And there are exonucleus and endonucleus sitting, they will chew up the mRNA as soon as mRNA enters the cytoplasm. So it needs to be protected, it needs to be shielded. And hence, in case of eukaryotes, we have two kinds of modification present at the two ends of the mRNA. One is known as the 5' capping. We'll do the details later on. And one is known as the 3' polyadenylation. So the primary transcript is protected, and next there is another modification that is done. The modification is done because DNA poses a problem in case of eukaryotic system. When we look into the gene of a DNA, so this is a transcription unit. This particular DNA that has been shown here is basically a transcription unit. In prokaryotes, we didn't face this problem because in prokaryotes, we didn't have structures like this, introns present. Now, in eukaryotes, most coding regions have intervening introns within them. What does this intron signify? It signifies that these sequences of DNA will not have a protein product. So there will be no protein. These sequences will not code for any protein. So if mRNA is the final product that will code the protein, how will a ribosome decide that we need to leave these introns out of the mRNA? So the nucleus has to ensure that before the mRNA leaves the nucleoplasm, the introns should be removed because they are not supposed to be present in the protein. So when we have this primary transcript, So we saw two modifications. One is a protection at the 5' end. One is a protection at the 3' end. And the third modification that is occurring are the removal of these brown structures from the mRNA. Because in the primary transcript, these brown structures, which were introns, were copied. As you can see, these introns were copied. But since the ribosome does not have a machinery to just jump, copy the exon, jump, again copy the exon. So we need to remove these introns. And you can see these introns are removed by a mechanism known as splicing. They're cut out from the primary transcript, and now we get a mature transcript which contains only the information of the exons. So this primary transcript now only contains the information of the exons. All the introns have been removed. So this is the mature mRNA. See, we never said this to be the mRNA. See, understand the title. This was said as primary transcript. This is not mature mRNA. But we are now calling this as mRNA. That means our introns have been removed. The mRNA has a 5' cap, and the mRNA has a poly A tail. The mRNA is protected. It is carrying the information of the exons only. And now the mRNA is transported to the cytoplasm. It is then captured by the translation machinery, and then it is decoded to a polypeptide. So the second difference you saw, the first difference between prokaryotic and eukaryotic transcription was that in case of prokaryotes, there is no nuclear membrane. In case of eukaryotes, we have a nuclear membrane. Second difference you saw is that in case of prokaryotes, there is no 5' protection given because mechanism is occurring within the cytoplasm itself. So there is no 5' protection given, and there is no 3' protection given. And finally, which is given in case of eukaryotes. And finally, another difference we saw in case of prokaryotes is that the primary transcript contains only the coding information of the DNA, which is directly decoded into the polypeptide. But in case of eukaryotes, the primary transcript contains both the coding and the non-coding information of the DNA. The non-coding informations are referred to as introns. The introns are removed to convert it into a mature mRNA, and then the mature mRNA is allowed to leave the nucleus, enter the cytoplasm, where it is decoded into the polypeptide. So conceptually, these are the two basic ways in which a prokaryotic transcription machinery differs from a eukaryotic transcription machinery.